which I was introduced actually by Stephen, so that was really nice. And then we met in America already 2 weeks ago. Matt comes also from psychology, studied in Michigan, and then we did a PhD at the New York State University in Stony Brook.
Yes.
Stony Brook. And is now working at the Control Lab in Georgia, which is a brave name, I think, for working with cinema and movies and engagement. Yeah, I'm happy to hear that now.
Okay, thank you, Katja. So I'm going to be talking about some different ways that psychologists and neuroscientists have looked at the effect of movies. So when we're talking about engagement, very simple, non-controversial definition that it's involving our cognitive and emotional mental processes as we are experiencing the world of a story. And this feature, along with some others, forms the theory of narrative transportation. As Steve was talking about these different related concepts to what we call engagement. And the way that psychologists and neuroscientists have looked at the effect of narratives has taken the assumption that if viewers are performing some of these same mental processes, we might see some similarities in the responses across viewers, and so they've looked for synchrony between them. Also, as Steve pointed out, we can look at first on a behavioral level of what viewers are doing. So as you watch this from the trailer to Ice Age, try to pay attention to where you're looking as you view the clip. Okay, so in research by the DM project, Tim Smith and others have actually had participants view this while they tracked where their eyes were fixating as they watched it. And so we can see here videos like this. Here, each of these green dots is a different single participant where they're looking. And this sort of color heat maps appear where there's tight clustering of where that eye gaze is focused. So you can see as you watch through this, see if it lines up with what you experience or your perception of that. What you can see in general is that there's this high degree of attentional synchrony where when you're viewing a narrative film, people tend to look at the same object. And as Steve pointed out, a really good predictor of where people will look is where there is motion on the screen. So we're seeing a high degree of behavioral similarity, and those eye gaze locations are the input that's going into the brain. So we already see a high degree of similarity there. From that input, we have the question of, once that enters the brain, to what extent are viewers' brains doing the same thing? This clip should look familiar because you just watched it in Steve's talk. The work of Yuri Hassan at Princeton has looked at the degree of similarity across brains using the beginning of the good, the bad, and the ugly. Okay, so using that type of clip, there's this question of when multiple people are viewing that, to what extent are their brains lining up? And our method for measuring brain activity, there's many different ones, but I'll be talking about fMRI. functional magnetic resonance imaging. And the basic logic of it is that neurons in particular brain regions will fire in response to particular tasks that they are recruited for. As a result of those neurons firing, oxygenated blood returns to that region to replenish them, and that's actually what we can pick up with the MR scanner. And then from measuring that blood flow, we convert that into colors on a structural brain image. So you'll see pictures like this with lovely colored blobs. These are just different slices of the brain, across, flat across, or cut up like bread, or through the middle. In black and white we have a structural brain image, and on top you see warm and cool colors. And typically in response to a task, the warm colors are areas that were more active for that task. The cool colors are ones that were less active. Another feature that's important for this synchrony work is that we also get a time course, a raw time course, of each point in the brain. This is from Yuri's work. So for example, this point in the visual cortex, there's a particular cube there that we're measuring, and we can plot the increases and decreases in signal in that region over time. Now, how we measure intersubject correlation, we take that same point in one brain and try to find it in another brain, and then these two time courses we see to what extent do they line up. And we can do that for all of the points in the brain. This figure illustrates one in the visual cortex, one higher up. But we repeat the process for the whole brain, and then we just see to what extent is activity correlated and where is it correlated. So for example, here's one particular brain region called the fusiform face area, which was discovered to be recruited for the processing of faces. So if we look at the activity across subjects in this brain region as they're viewing the first half hour of The Good, the Bad, and the Ugly, you can see, even just looking at the graph, that there's a high degree of similarity in the peaks and valleys on this function across subjects. And if we look at what's happening at those peaks, here we have a figure of the correlation across time for this particular region. And at the top peaks, the common feature is that people are processing faces in those scenes. And we can contrast this with other regions that are recruited for other tasks. Okay, so looking across the whole brain, how much of it is synchronized when people are viewing Good, the Bad, and the Ugly? They found that about 45% is correlated. So a decent percentage of the brain. Now, you might ask, like, is this just any processing of a video that would do this? Is there something special about narratives? So we can compare this to other videos that we might not expect to engage these same cognitive and emotional processes. So here's just a video of Washington Square Park in New York City. And Hassan and colleagues didn't use this video, but a similar video of Washington Square Park, which may have been even less engaging because I think they used a static camera. But you can see it's very complex audio and visual input, but there's no story, and there's a lot of action happening on screen. There's not really a focused narrative of what to look at. So if we compare the degree of intersubject correlation between these two, what we see is that both The Good, the Bad, and the Ugly and this video of the park are activating a high degree of correlation in visual and auditory primary processing regions. So the regions that first process our sensory input are aligned across viewers, but only the narrative film extends in this blue region, which goes beyond basic processing to presumably more higher order processing. So there's a higher degree of intersubject correlation, and the difference here is that we would expect that there are these higher order processes occurring when they're viewing the narrative film. And there's a difference between these two. Yuri has looked at a few different films. There's a clip from a Hitchcock television program, The Good, the Bad, and the Ugly, episode of Curb Your Enthusiasm, and then that amateur video of Washington Square Park. And you can see that the movies vary in the degree to which they elicit this intersubject correlation, suggesting that this is capturing some difference in the level of engagement between these. And it's only 4 examples, so we can't really systematically say what are the features that cause the highest correlation, but we could see that there are differences between different types of films. Okay, and it's important to note here that the strongest correlations we see are in those auditory and visual processing areas. However, in narrative engagement, we're really interested in these much more interesting processes like mental participation, the emotional responses, the thoughts that we have about the characters, and these are assumed to be carried out in using the frontal lobes of the brain, which, as you can see, we don't see a lot of correlation there. So there's a question of, could we be missing some sort of synchronized activity between the participants due to just the increased anatomical and functional variability between participants that we have in these more advanced regions? So to get at this question, there's a new technique developed by James Haxby and colleagues called hyperalignment, which might improve aligning participants better than we can just by lining up their brains. So traditionally, we just align brains by trying to match the anatomical space. So you have two different brains, you try to warp the shape of them so that they match up as closely as possible. You align them to a model, and then you try to say the dot on this brain corresponds to this dot on the cortex of the other brain. However, doing this type of alignment is actually very bad at predicting the activity from one brain to the next. So within a particular participant, we could train a classifier to be able to predict the activity for a particular stimulus, but if you use that model to try to predict the activity that you'll generate in another participant, it performs very poorly due to this slight differences in the wiring between the brains. And the new contribution of this hyperalignment technique is that it uses feature films to align brains not based just on anatomical space, but on the common representation of what people are experiencing in the moment. So I won't go into too much detail of this method because it's very complicated. But over here on the left, they had participants view the film Raiders of the Lost Ark, and they took each moment in that film as a particular stimulus. And then they showed it to a series of subjects, and so you can see with the subjects, say for this moment in particular, it generates a pattern, a grid of responses in different voxels, at different points in the brain. Some are more or less active. Now, by the traditional anatomical alignment method, we would just try to line up each point in the grid to each other, and you can see that there may be some correspondence there, but there are also differences. So just as an illustration, these three different voxels, we can map that in space and see that for these different points in the movie, they would appear in different points in the space based on the different responses there. So what hyperalignment is doing is saying we can transform the grids to try to align them not on the anatomical representation, but to try to match what's happening at the moment. So if there's a common representation of this particular point in the film, we can change the space of the grid in order to line these up. And up on the top here we see, this is just a replication of the same pattern we see with Yuri Hassan's work, where we're aligning brains anatomically, trying to match the physical space as much as possible. And here we have alignment of about 45% of the cortex is correlated. However, after performing hyperalignment, then we see during the movies that we watch, there's alignment of 75% of the brain. And we even see recruitment in some of these frontal regions, which we would expect would be involved in narrative engagement. So this is an improvement. Now it's an open question as to what this alignment means. So we know that this is based on a common representation of a particular point in each film. And then it's an open question as to whether this reflects that the different brains have the same emotional state, or whether it's just different responses but to the same point in time. And it's also important to emphasize that even with this technique, we're only seeing 75% alignment. So it's not that viewers are having the exact same experience. There's clearly going to be very interesting individual differences. But this is just showing to what extent there is a commonality between them. And to get at what are the contributions of these cognitive and emotional processes, it really requires more targeted experiments. So I'd like to get back at the idea of narrative transportation, because this is where I've conducted a lot of work concerning attention. So according to Richard Gehrig's theory of narrative transportation, we're devoting our cognitive and emotional mental processes to the world of the story. And as a result of this process, we're not devoting these processes to our physical environment, so aspects of the real world are less accessible. We don't notice sounds or other things occurring in our environment. We don't access knowledge from memory that might be inconsistent with the world of the story when we're in the moment of viewing. And also, the experiencer might be changed as a result through the formation of new emotional memories or through attitude or belief change to views that are more consistent with the world of the story. In my work, I've looked particularly at this middle part of a loss of attention to the physical world. And specifically, I've looked at suspense as a particular type of narrative device that we can make predictions about what effect it will have on attention. Suspense arises with threats to characters. So when narrative content emphasizes that there may be a threat to a character, we experience suspense. It's a mixture of hope for a positive outcome and fear for the negative outcome. So it's an interesting mixture of these positive and negative emotions. And some work has shown that if narratives are framed to increase suspense, to focus on potential threats to characters, then participants report increased transportation on the kinds of self-report scales that others were discussing earlier today. Okay, so my specific attention hypothesis of suspense deals with what's happening on a moment-by-moment basis in the films. And the hypothesis is that at moments of increasing suspense, we'll find increased narrative-related processing and decreased non-narrative related processing. And this is a very broad hypothesis that could be tested in many different ways. In some other previous work, I've used dual task procedure like Stephen was talking about. Today, I'm going to be talking about using an fMRI paradigm to test this question. So to separate narrative and non-narrative processing, we're going to actually test for the theory that suspense will narrow attention. So if we present narratives in the center of where people are fixating on and have other stimulation in the periphery where they're not focusing on, can we see a difference as a basis of the level of suspense in a film? We predicted that at time points where suspense is rising, participants are going to show decreased processing of that visual periphery. And then we also predicted that there would be downstream consequences of this focused attention, such that if we give a memory test afterward, participants will have improved memory for suspenseful time points relative to other ones in the film. And the way we're able to take advantage of the architecture of the brain to study this question is due to the way that the primary visual cortex is organized. This is the first part of the brain where visual processing takes place. And it's one of the areas of the brain that we understand the best. So the point in your visual field that you're directly looking at, on the right here we have a visual field, the part you're fixating on, that's processed in the very, very back of the calcarine sulcus in the brain. And then everything else around the periphery is processed further forward. So it's not really important for our purposes, the very specific mapping. The mapping of the world is preserved in the brain, but for our purposes, the very important thing will be that we can map out these separate center and periphery areas. So in our experiment, we have a checkerboard that flashes either in the center or in the periphery. And this stimulus generates a lot of activity in those particular regions so that we can map out the center and the periphery and then test this theory that attention will narrow. So after mapping these out, we presented a somewhat unusual format for watching movie excerpts. We had suspenseful film clips in the center of the screen and then continuous flashing checkerboards around the border. And participants were instructed to keep their eyes on the center of the screen, but otherwise try to pay attention to the movie clips. It obviously is not the ideal viewing conditions, but in the interviews we gave afterwards, it seemed that participants were able to focus on them. And we did have sound with the film clips as they watched them. We used a series of clips from 10 different films, some Hitchcock films. These spanned a very wide range of time and were different in the specific content that they had, but they all had the same feature that they had a character facing the threat of a negative outcome that would increase and decrease over the course of about 3 to 4 minutes as a function of the emphasis of the threats on those characters. So for an example, and for an interactive point, in this clip from North by Northwest that you probably have all seen, I would like you to watch this, and at any time point where you perceive a threat to the character, just tap on the table or on your lap, and then pay attention to when those moments are. Okay, so that guy can't catch a break. All right, so you may have noticed that at several time points there you were noticing threats to the characters. So we had participants give ratings, and we didn't use just the knocking procedure, which I'm going to copyright, so nobody take that. We had a slider on a scale that people could move up or down to indicate how strongly threats were emphasized. We used the perception of threats to try to specifically operationalize what we were looking at, because people might have different definitions of what they consider suspense or engagement or presence. So in this experiment, we were just looking at that as our slider measure. But other studies could look at all sorts of other different measures. And if we look at the average response across participants, this is the one for that scene from North by Northwest, we can see there's actually a high degree of agreement between participants, and these peaks correspond to meaningful content in the film scenes, the moments when the plane was swooping and attacking Cary Grant. Now, across our 10 clips, the actual threats to the characters were quite different, so it wasn't always a similar feature of what was happening, but it was linked in that there was an increased threat. Now, what we did with our MRI study is we took those ratings that participants had made when suspense is increasing or decreasing, and we applied it to the data of when they're viewing the clips with the checkerboards to try to see if there's a difference in the processing of the periphery as a function of suspense. Now, we're interested in suspense, but there's also a lot of low-level features that are fluctuating over time in the movies. So we just wanted to control for some of these color properties, edges, and the motion between frames because we want to take them out of the regression model so that we don't include the effects of suspense. They're not taken up by these confounding variables. We also gave a memory test, and this is from a similar related study to the previous one, but I'm just concatenating it here for clarity. We had participants answer questions for an event that happened in each 20 seconds of film time. So for example, one question from this one is, when Roger tries to flag down the truck, what happens? Anybody remember?
He almost gets run over.
Yes. So that would be a correct answer for that one. And we were interested to see if memory is better for high suspense versus low suspense moments. So as I said before, we can map out brain regions that process the center and the periphery. So here we have the results plotted on a inflated brain surface. Up here, this is a view from the inside on the left and right. And then here, this is a view on the outside if you were to split the brain open. And we're particularly interested in what was happening in that region that processes the visual periphery, because our hypothesis is that due to attention narrowing at suspense, we'll see less activity in the region that processes the periphery. So what do we find? Our hypothesis was confirmed. That area was less active, and there was more activity in the region that processes the center, suggesting that there is an attentional narrowing on a moment-by-moment basis within suspenseful film clips. Some of the other results that we also found with suspense, so here warm colors were more active at high suspense moments, cool colors less active. We see more activity in these higher order visual processing areas. More activity in the ventral attention network, which is associated with processing engaging stimuli, particularly from a bottom-up direction. I know Steve was talking about top-down and bottom-up influences. This suggests that it's driven by the narrative at these moments. So when a new threat appears, attention is directed to the clips. And we see less activity in some of the nodes of the default mode network, which was identified as brain areas that were more active in the absence of task engagement. I know you've done previous boot camp on default mode, so you guys probably know even more about it than me, but its function is not simple, but it's involved in many different internally directed tasks, such as mind wandering, theory of mind, moral decision making. But all these tasks share an inward focus of attention, which we would expect to be less engaged at moments of high suspense. Okay, so our memory task, we had independent raters score each participant's answer with a high degree of agreement between them. And then we split those questions based on whether they came from high or low suspense events. So we can compare memory on the basis of the suspense at each of those points in time. And we predicted that performance would be better for questions that came from those high suspense moments. And our results confirm this. Participants were much better at remembering the events at high suspense moments. But we can also look at what was happening in the brain during time points that would later be correctly remembered. So for this analysis, we compared time points for each participant that they later correctly recalled minus the brain activity during time points when they did not correctly recall activity. So here I have plotted the views of the outside of the brain, the inside, and then this bottom one is a view looking underneath. You can see there's a significant degree of overlap here with our suspense results, which makes sense because suspenseful moments are better remembered. But we also see activity in the medial temporal regions here, which have previously been implicated in memory encoding. So this supports the idea that these high suspense moments are better encoded in memory. So just to summarize, we see that suspense, when it's increasing within a suspenseful excerpt, it's causing participants to suppress processing of the periphery and boost their central processing. And we also see recruitment of other attentional and visual processing regions, deactivation of the default loop network. So this is clearly an externally focused attention, but specific to the narrative and not the non-narrative related periphery. And participants have better recall for film events from those high suspense moments. So narrative engagement involves these cognitive and emotional processes, and neuroscientists have shown that films vary in the extent to which they evoke synchronized brain activity and the extent to which that covers the extent of the brain. And by investigating these processes on a moment-by-moment basis with specific predictions about how they will affect attention, we can reveal how they unfold over time. And then a final point I want to make is that we didn't evolve to watch films. Films have evolved to take advantage of our processes. So they're taking advantage of these processes we use to adaptively interact with the environment. And so by using films, we can study the way attention and memory processes in everyday life work using a medium with a high degree of realism. So I'd like to thank my collaborators and thank you for your attention.